Hi dear students. So far we completed first four chapters which are the part of chemistry. Now we are moving to the section biology. Here we are going to start with the chapter number 5. In this video I am going to discuss the first chapter from biology, that is the fundamental units of life. This chapter is actually dealing with the structure and details of the cell. In class 8 also we have learned basic idea of cell, what is a cell and basic components of the cell. The first thing we are going to discuss in this chapter is that some historical aspects related to the discovery of the cell. The next, microscope, and we will discuss what is cell and some characteristics of the cell, and on the basis of cell there are two more topics we will discuss, that are multicellular and unicellular. Then we will move to the parts of the cell. There are four main types of parts. First one is nucleus, plasma membrane, cell wall, cytoplasm, and cell organelles. Then we will discuss different cell organelles: endoplasmic reticulum, Golgi apparatus, lysosome, mitochondria, plastids, vacuoles. And again, endoplasmic reticulum divided into two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. And plastids also different types: chromoplast, leucoplast, and chloroplast. These are the types of plastids. And the last topic in this chapter we will discuss, that is cell division. Cell division also there are two types. First one is meiosis and next one is mitosis. So these are the topics we are going to see in this chapter. Out of these all topics, today I am going to discuss up to here, up to the basic parts of the cell. Okay, the cell organelles, different types of cell organelles, and cell division. These two topics I will discuss in the next video. So let's start with the discovery of the cell. In 1665, the scientist named Robert Hooke took a slice of cork and observed under a self-designed microscope, which is actually a simple magnifying lens. And he saw that the cork resembled the structure of honeycomb containing little compartments, and he had called each compartment as a cell. So the name cell is given by the scientist Robert Hooke. We know that cork cells are dead cells which are obtained from the outer layer of tree. So he observed dead cell. After his discovery in 1674, Leeuwenhoek observed the first living cell under the microscope. That means Leeuwenhoek was the person who discovered the living cell. So these are the basic things you have to remember regarding the discovery of cell and the further development in the discovery of the cell done by different scientists, like a discovery of nucleus, cytoplasm, etc. So this whole part is given in your textbook, so just read that. Now let's see what is a compound microscope. Because Robert Hooke used simple microscope which consists of single lens, and nowadays we are using a sophisticated microscope called compound microscope. It consists of two lenses. What is compound microscope? An optical instrument for forming magnified images of small objects, consisting of an objective lens and an eyepiece. Both lenses are mounted in the same tube. So that instrument is called compound microscope, to get the magnified image of small objects like cell and microorganisms, etc. So let's see what are the parts of the compound microscope. This is a typical compound microscope. This is the mirror in which the light is incident on this mirror and it reflected back and it passes through the body tube. And this is the stage in which we will keep the slide for observing. And this is the clip for mounting the slide which we prepared, or the permanent slide. And this is the objective lens of different power. This is 100, this is 40, and this is 10. We can change the power of this objective lens by rotating that knob. You can see here, if you choose 40, the 40 will be along the body tube. If you change it to 10x, then the length of power 10x will become along the body tube. And this is the body tube. And this is the coarse adjustment. And this is the fine adjustment. When we twist the coarse adjustment, you can see the body tube will be moving upward, and if you twist in the opposite direction, it will move downward. So to adjust the focus. Similarly, this is the fine adjustment. Fine adjustment is for just adjusting in a small variation. So that cannot be seen from outside. That will be adjusted inside the body tube. This is the eyepiece through which we observe the magnified image of the specimen. So this is all about the basic structure of compound microscope. Now we are going to discuss what is a cell. We can simply say that cell is the structural and functional unit of life. It is a structural as well as functional unit of life. Why? All living organisms are made up of cells, and a group of cells form tissue. And further, the group of tissues to form organ, then organ system. And different kind of organ system contribute to form an organism. So structurally, cell is the fundamental or structural unit of the life. Now how it is become functional unit. Also, each cell having the capacity to perform certain basic function. And for example, living cells secrete enzymes and hormones which helps for digestion. Each cell have the capability to perform their unique function. Because of that, cell is also called functional unit of the organism or life. So this is the thing we can say about what is a cell. Cell is not a single substance which is composed of different components called cell organelles. We will discuss different type of cell organelles in detail later in this chapter. I told you that different cells perform different functions. Because of that, different kind of cells or different type of cell have different size and shape. It depends on the function in which they perform. It doesn't depend on the size and shape of the organism. For example, the nerve cell present in rat and elephant both are elongated in the structure because the function of the nerve cell is to transmit the stimuli from one part of the body to the another part. That means it doesn't depend on the size and shape of the organism. It depends on the function in which they perform. You can see here different kind of tissue cells present in the human, that is nerve cell, blood cells, muscle cells, spindle shaped muscle cell, ovum, sperm, bone cell and fat cell. So these are the different types of the cells. All the organisms are not made up of same number of cells. It depends on the function they perform, depends on where they live, and depends on the size of the organisms. The number of cells present in their body are also different. And on the basis of number of cells present in the organisms, they are classified into unicellular and multicellular organisms. Uni means single. So unicellular means the organism which is made up of a single cell is called a unicellular organism. For example, bacteria, paramecium, amoeba. These all are examples of unicellular organisms because their body consists of a single cell. On the other hand, some organisms which are made up of millions or trillions of cells like us. So such types of organisms are called multicellular organisms. Multi means more than one. All the animals, plants, etc. are examples of multicellular organisms. And one more thing we need to remember. In case of multicellular organisms, different part of the body perform different functions. While in a unicellular organisms like an amoeba, all the functions, locomotion, digestion, food capturing, all these functions are performed by a single cell. This is the main difference between unicellular and multicellular organisms. So now let's discuss the parts of the cell. If we look in detail into the cell, we can observe that cell is composed of different parts. The first one is plasma membrane. Second one is cell wall. That is also not present in all type of cell. It is only present in plant cell. Then nucleus, cytoplasm, then cell organelles. Out of this, plasma membrane, nucleus and cytoplasm, these are present in almost all the cells. That is the basic structural organization of the cell. And well-defined cell organelles are present only in case of certain type of cells. And that is also different in different type of cells. Some cell organelles present in some kind of cells, some are absent in another type of cells. Similarly, cell wall. Cell wall is not present in animal cell, it is only present in plants. But plasma membrane, nucleus and cytoplasm. Plasma membrane is just an outer boundary of the cell. Nucleus is the controlling coordinating part of the cell. And cytoplasm is the region between nucleus and plasma membrane in which cell organelles are embedded in it. Okay, so this is the basic three parts of the almost all the cells. Now let's discuss one by one. First one is plasma membrane. The plasma membrane is flexible and it is made up of lipids and proteins. This is the boundary of the cell, all cells, and it is flexible. Let's see the structure of the plasma membrane here. See, this is the outside area of the cell and this is the inside. So this is plasma membrane, okay? And this yellow structure, small yellow structures is lipid, okay? Two layers of lipid, and this red structure, that is protein, through which nutrients and other molecules is transported. Details of the structure of the plasma membrane is not in your syllabus, okay? So just understand, this is a flexible and it is made up of lipids and proteins. These are the two main components of the plasma membrane. And it is also called a selectively permeable membrane or semi-permeable membrane, because plasma membrane allows the passage of certain types of materials in and out of the cell. On the other hand, it prevents the movement of certain materials. That is, only certain types of materials can pass through the plasma membrane. Because of that, it is called a selectively permeable or semi-permeable membrane. As it is a selectively permeable membrane, the substances like CO2 and oxygen can move across the cell membrane by a process of diffusion. Another name is cell membrane, okay? Sometimes you will call it a cell membrane. We can call it is a plasma membrane and a cell membrane, then selectively permeable membrane and semi-permeable membrane. These are the four names we can call it for. So the substances like oxygen and CO2 can move through the cell membrane by the process of diffusion. What is diffusion? We already learned that diffusion is the intermixing of particles on their own. Or we can define in another way that movement of particles from the region of higher concentration to the region of lower concentration spontaneously. That means without application of any external force or pressure. The same way, exchange of oxygen and carbon dioxide gases is done. How it is? Sometimes when the impurities and the carbon dioxide become more inside the cell, then the concentration of carbon dioxide become high in the cell compared to the exterior of the cell. That means the carbon dioxide will move from the region of higher concentration. That means from inside the cell and to the outer part of the cell. That means carbon dioxide is the waste part which will be removed from the inner part of the cell to outer part by the process of diffusion. The same way how oxygen will be entered into the cell. Once the respiration is done, then the concentration of oxygen in the interior of the cell will be less compared to the outer part of the surroundings of the cell. Then the oxygen molecules will move from the region of higher concentration, that means from the surroundings, to the inner part of the cell, because the plasma membrane permits the movement of oxygen and carbon dioxide. So the property of plasma membrane, as it is selectively permeable membrane, is a great advantage for the exchange of carbon dioxide and oxygen. The plasma membrane also permits the movement of water across the cell membrane. So that process is called osmosis. That is, osmosis is the passage of water from the region of higher concentration through a selectively permeable membrane to a region of low water concentration till the equilibrium is reached. That means osmosis is a special kind of diffusion in the case of water if it passes through selectively permeable membrane. Say it is a diffusion actually because water is moving from the region of higher concentration to the region of lower concentration through the selectively permeable membrane. So if it is moving water through a selectively permeable membrane, then that type of diffusion is termed as osmosis. So osmosis is a special kind of diffusion. It is only we will use in case of water, and also normal diffusion of water also will not term it as osmosis. It should be take place through the selectively permeable membrane. That means like a plasma membrane. As I told you that plasma membrane is highly flexible. This flexibility of plasma membranes make an advantage in the unicellular organisms by the process of endocytosis. What is endocytosis? A process in which a cell takes in materials from outside by engulfing and fusing them with its plasma membrane. When their prey comes closer to the cell, then the cell will change its shape by making pseudopodia. In case of amoeba, amoeba is the best example we can give in which an organisms capture food by endocytosis. So it changes its body shape and produce, and it makes some projections outward, that is called a pseudopodia. And when the food particle enters inside, it will close and it will engulf the food. This is possible only because of the flexibility of the plasma membrane of these unicellular organisms. So this is all about, you need to understand what is plasma membrane and the terms related to plasma membrane or selectively permeable membrane. I told you that osmosis is a special kind of diffusion in which movement of water takes place from the higher concentration region through a selectively permeable membrane to the lower concentration region. Let's discuss the osmosis in detail. We are going to discuss a situation that if an animal cell or plant cell put in a sugar or salt solution, there are three possibilities we have in the case of concentration of the solution. Okay, and according to that, the direction of the osmosis will be different in different situations. So that let's discuss in detail. First one is hypotonic solution. What is hypotonic solution? Hypotonic solution means the concentration of water in cell is less than concentration of water in the surrounding medium. That means the solute content, whether it is salt or sugar, is less in that water. So we can call that as a dilute solution. Okay, so in this scenario we will call it as hypotonic solution. And in case of concentration of water in the cell is less than that of the water, what will happen? What do you think? According to the osmosis, the water will move from the region of higher concentration to the region of lower concentration. So the overall movement of water will take place into the cell, even though in both direction movement of water will take place. Net movement will be from outside of the cell to inside of the cell, because inside the cell the concentration of water is less than that of the outside or in the surroundings. Okay, so overall movement will take place into the cell. Let's see here. And when this overall movement of water take place into the cell, what will happen? The volume of the cell will increase as the water enters into the cell. How volume can be increased? Because the plasma membrane, that is the membrane of the cell, is flexible. So due to its flexibility, it can bulge out, or we can say that it is likely to swell up. So that is, in this picture you can see that this is the blood cell. Actually this is swell up because of water molecule is entering into the cell. You can see the solute concentration of the solute particle is less, which means concentration of water is high compared to the concentration of water inside the cell. So water will enter into the cell, so that cell will become swollen. You may already observe that when the pulses and the grams are soaked in the water for a couple of hours, then you can observe that it is swollen. This is a real life example we can give for this hypertonic solution, in which osmosis takes place from surrounding into the cell. Second one is an isotonic solution. In case of isotonic solution, that is concentration of water in the cell is equal to the concentration of water in the surrounding medium. So in that case we will call isotonic. That means concentration is same. Iso means same. Okay. So in that case, what will you think? If the concentration in the cell as well as in the surrounding is same, then of course some movement of water will take place, but the movement of water from cell to the surrounding and from surrounding to the cell will be equal in amount. So therefore, no overall movement of water. That means cell is likely to stay in the same size. You can see the concentration, movement from the surrounding to the cell as well as from cell to surroundings will be take place in equal amount. So obviously there is no overall movement or net movement. So the cell is likely to stay with the same size. The third possibility, that is hypertonic solution. What a hypertonic solution? The concentration of water in the cell is greater than the concentration of water in the surrounding medium. That means the surrounding solution, if it is salt or sugar, the amount of solute is more, which means it is concentrated solution in terms of the solute. Okay, that means in terms of sugar or salt. That means concentration of water is less compared to the concentration of water in the cell. So in that case, we will call that as a hypertonic solution. So in that case, what do you think? Obviously, the water will move from cell to the surroundings. Obviously from surrounding to cell also, but overall movement of water will take place from cell to the surroundings. Overall movement of water into the surroundings from the cell. Because the solute particles are more, which means concentration of water is less. So water will move from the cell. As a result of this movement of water from the cell to surroundings, what will happen? Cell will get a shrink. So the cell will shrink. This is the third possibility. From real life, we can take an example of this salted mangoes. If mango, without breaking into pieces, keeping in salt solution, highly concentrated salt solution, that means concentration of water is less than that of water present in the mango. For a long time, you can observe that the mango will get shrink. It is because the osmosis takes place from mango to the surroundings or to the salt solution present in the wood. This is the real life example you can get for this hypertonic solution. Some examples, activities given in your textbook, you can also read that one. I told you that plasma membrane is essential for all the cells because that is the boundary of the cell. Okay, so in plant cell, in addition to plasma membrane, there is one more layer, a rigid layer is called cell wall. The rigid outer covering present in the cell, plant cell, is called cell wall in addition to plasma membrane. That is very important. So you can see here, this is the structure of a typical plant cell. This yellowish area, this layer, the first layer, inner layer, that is actually plasma membrane. We have discussed. On the plasma membrane, there is a rigid green layer. You can see, actually it is not necessary to be green. Okay, this is just a structure only. That rigid layer is called cell wall. This is only present in plants as well as in some bacteria for supporting the cell. Then how this cell wall is become so rigid? The reason is that it is composed of cellulose. Cellulose is a complex substance and provides structural strength to plants as it makes a layer called a cell wall around the plasma membrane. We know that cellulose is a natural fiber or polymer which is made up of glucose. Because of that, it has become rigid and it is provided structural support for the plant cells. And one more term related to this, you have to remember that plasmolysis. Plasmolysis is a phenomenon, it's a process in which the shrinkage or contraction of contents of the cell away from the cell wall when a living plant cell loses water through osmosis. If a cell is kept in hypotonic solution, the cell will lose the water. As it loses the water, the content present in the cell as well as the plasma membrane will get shrink. But the shape of the cell wall will keep same because it is rigid. So what will we observe here? You can observe that this content, the entire content, plasma membrane including all the contents will get shrink. That means it take away from the cell wall. Cell wall will keep remain its shape. So that is called the plasmolysis. That will happen in case of plant cell. So that means you can say that even if there is a lossage of water also, the cell content will shrink, but the space which is required to keep that cell will be same because cell wall is keeping its shape is same. That is, cell wall helps the plant cell to withstand much greater change in the surroundings than animals. So if there is any change in the concentration of surroundings, what will happen? Cell wall will provide that support. Plasma membrane cannot because it is highly flexible. This is not only for losing water by osmosis. We know that plants are immortal. That means they are exposed to the various changes in the climate like temperature, pressure, and they are exposed to heavy rain, wind, etc. So they need to get some strength to prevent or to protect their body from these changes. That means the body of the plant should be much strong than the animals. This strength is provided by the cell wall. So cell wall is only present in plant cell. Why? Because of these reasons. Okay, so this is all about the cell wall unit on this. So now we have discussed the first structural part, that is plasma membrane, in addition to that cell wall that is present in only plant cell. The second important part, that is nucleus. It's a controlling and coordinating part of the cell. So it is an important component of the living cell and it is generally spherical and located in the center of the cell. Not always, generally, not necessary, always. Let's see the structure of nucleus. See here, this is the typical plant cell. You can see here the spherical part, that is nucleus. And it is generally located in the center but not necessarily always. And is one of the important component of the cell. That is you have to remember. Okay? And there is a membrane, double layer covering of the nucleus is called nuclear membrane. And it has pores which allows the transfer of material from inside the nucleus to outside it. There is a membrane of double layer. That is called a nuclear membrane, which is covered by the nuclear materials present in the nucleus. And this layer has a lot of pores. This is the first part of the nucleus, that is nuclear membrane. Like a plasma membrane is the boundary of the cell, nuclear membrane is the boundary layer of the nucleus because it is a well-defined part of the cell, because it is the centermost part or it is a controlling and coordinating part of the cell. Now let's discuss the other components present inside the nucleus. First one is chromosomes. Chromosomes, they are thread-like structure present in the nucleus. We can discuss in detail, that is, in a cell, if it is not dividing for the growth, then the chromosomes are present as entangled mass of thread-like structures called chromatin material. You can see here, this is the cross-section of the nucleus. And there are a lot of thread-like structure. This is called a chromatin. During the normal time of cell, the chromosomes are present as chromatin, which are thread-like structure. But whenever the cell is about to divide, the chromatin material get organized into chromosomes. That means it is organized into the rod-like structure that is called chromosomes. This is the way how chromosomes. So this structure of chromosomes, rod-like structure, will be seen only during the division of the cell, when cell is about to divide. Otherwise, it is present as chromatids. That means chromosomes are composed of chromatids. During the time of cell division, this thread-like structure of chromatids will get organized and form rod-like structure. That is chromosome. So, chromosome is composed of chromatids. And again, it is composed of DNA, that is deoxyribonucleic acid, and protein. So, chromosomes composed of chromatin, and that chromatin is composed of DNA. Its full form is deoxyribonucleic acid, as well as it is composed of some proteins. DNA structure is highly complex. It is actually double-stranded helix structure. This is the typical structure of DNA. And this DNA stores, or it contains information for inheritance of characters from parents to the next generation. Different characters which we have that are transferred from our parents. So the inheritance of characters from generation to generation, that is actually done is because of the information stored in the DNA. And also it is for constructing and organizing the cell. The important information for constructing the cell and organizing the cell. This all information are stored in the DNA. And each DNA present in the cell of different organisms, especially in case of human being, are unique. This is the reason nowadays we are using DNA fingerprinting method to identify the person. If we get a blood sample or hair or something from our body part, this whole body part consists of cells and DNAs. By using this DNA fingerprinting, it can be identified the person. Because the sequence of biomolecules in the DNA are unique in every individual. And also, this DNA again composed of functional segments called genes. So the structure of the chromosomes are highly complex, composed of chromatin, and the chromatids are composed of DNA. Again, this DNA is composed of functional segments called genes. Again, this DNA composed of different types of biomolecules. So it is highly complex in nature. It requires high resolution electron microscope to observe these structures from a nucleus. We already said that chromosomes contain these information. So the number of chromosomes present in an organism determine its complete characters. For example, in humans, the number of chromosomes present that are 46. Similarly, in different organisms, the number of chromosomes are different. Okay, so this is all about the structure of the cell. Two main things. First one is nuclear membrane. It's for separating the nucleus from the remaining part of the cell. That's a boundary. Second one is chromosomes. That is the main content present in the nucleus. And on the basis of this structural organization of nucleus, the organisms are classified into two categories that we are going to discuss. Not on the basis of structural organization. Entire cell also okay, but mainly on the basis of nucleus organization or how nuclear material is organized. See, there are two pictures we have. First one is the cell of the unicellular organisms. It is a cell of a bacteria. And this is the structure of a plant cell. So you can see here, this is a unicellular organism. There is a separate well-membraned nucleus, but here there is no separate well-membraned nucleus. This nuclear materials called nucleoid is distributed undefined. So this is the main difference you can see here. That means in the multicellular, especially in the multicellular organisms, the structure of the nucleus and the other part of the cells are well defined. While in case of these unicellular organisms. The structural the nucleus is not well organized. That is the main difference. Not only nucleus is the one of the factor. In case of other, you can see here ribosomes are the plasma membrane and cell wall, nucleoid, and these materials are distributed randomly. But here there is a separate membrane. These cell organelles present. These are the different cell organelles. We will discuss later in detail one by one. Okay, so on the basis of these differences, the organisms are classified into prokaryotes and eukaryotes. Prokaryotes means the organism with the prokaryotic cell, and eukaryotes are the organism with the eukaryotic cell. What are the main differences? In case of size, generally small and 1 to 10 micrometer. 1 micrometer, this is representing micro, mu. Symbol is mu. Mu. This symbol is actually mu. Okay. It means micro. Micro means 10 raised to minus 6 meter. So 1 to 10 micrometer, and the size of eukaryotic cells are generally large, 5 to 100 micrometer. And in case of nuclear region, undefined nuclear region containing only nucleic acid and nucleoid. That is not well defined, is somewhat randomly distributed throughout the. And here nuclear region is well defined, surrounded by a nuclear membrane that is completely separated from the remaining part of the cell. And in case of chromosomes, single chromosomes. Here more than one chromosomes. Like I told you that in case of humans, there are 46. And in case of cell organelles, membrane bounded cell organelles are absent. All the cell organelles are randomly in the different part of the cell, and there is no membrane for cell organelles also, like in case of nucleus. While here in case of eukaryotic cell, membrane bounded cell organelles are present. That means we are going to discuss the different cell organelles later, but I will tell you some cell organelles like for example plastid or mitochondria. Mitochondria is a cell organelle present in the cell. In case of eukaryotic cell, that is covered by a membrane. Similarly, all the other cell organelles. That means all the different types of cell organelles are separated from the remaining part of the cells by a membrane. So this is in case of the eukaryotic cell. And examples of bacteria, here animals. So this is the two types of organisms on the basis of the organization, structural organization of the cells, especially in case of the nucleus. Now let's see the last topic of this video, that is cytoplasm. It's a fluid content between nucleus and plasma membrane. See here, this is the nucleus, okay, and this is the plasma membrane. Okay, and the region between the plasma membrane and nucleus, this is called cytoplasm. And also it contains many specialized cell organelles. You can see here, this is one kind of cell organelle, this is another kind, this is another kind. Okay, this is the another kind, and some small cell organelles also present. Okay, so different kind of cell organelles, well defined cell organelles are embedded in this cytoplasm. It is a fluid content. So except to plasma membrane, cytoplasm plus nucleus together called the protoplasm. Cytoplasm plus nucleus called the protoplasm. So this is all about the first part of this chapter. In the next part, I will discuss the cell organelles in detail. So that's all for today. Thanks for watching.